So what are we concerned with? We're concerned with breast cancer, and we have good reason to be. Breast cancer is estimated to affect 1 in 8 women in their lifetime. In fact, in the United States alone, in 2013, there were over 230,000 new cases of breast cancer diagnosed. Traditionally, breast cancer has been classified into 3 main subtypes: ER positive, PR positive, and HER2 overexpressing. There's also triple negative forms. So ER positive and PR positive are normally grouped together, then there's the HER2, and then a triple negative. Now 70% of those tumors are the ER or PR positive tumors, and they express a nuclear hormone receptor called estrogen receptor alpha. Fortunately, a drug called tamoxifen, which acts as a competitive inhibitor to estrogen with regards to the estrogen receptor, works quite effectively against these tumors, and it's become the gold standard for treatment of ER-positive breast cancer. Unfortunately, 30 to 40% of patients treated with tamoxifen develop resistance to the drug over 5-year courses of treatment. Previous studies have implicated the transcription factor EIF4E in the development of this drug resistance. So, what is EIF4E, and what does it have to do with our project? EIF4E is a transcription factor that is hyperactive in tamoxifen-resistant breast cancers. It selectively translates the proteins that help the cell to proliferate, even in the presence of tamoxifen. In a normal human cell, that is, a non-cancerous human cell, the protein 4E-BP1 binds to and sequesters EIF4E when it's unneeded. And once it's sequestered by 4E-BP1, EIF4E is unable to translate those oncogenic factors that promote the drug resistance. Now, that's all well and good until 4E-BP1 is unable to sequester EIF4E. And that happens when 4E-BP1 is phosphorylated. Any of you guys who have taken basic biology or basic chemistry knows that when you phosphorylate something, you're capable of inducing a conformational change. So, yes?
I'm sorry, what's the downstream effect of EIF4E?
The downstream effect of EIF4E, it selectively translates proteins that help the cell to proliferate even in the presence of tamoxifen. And do those questions count in my time? Because I thought those were at the end.
Just go.
So, mTORC1 is capable of phosphorylating 4E-BP1 in several sites and inducing that conformational change. That conformational change can leave 4E-BP1 unable to bind to and sequester EIF4E, thereby allowing EIF4E to kind of go rogue and translate those oncogenic factors. Our project wants to better understand this signaling access by purifying and isolating wild-type 4E-BP1 and EIF4E. But we also want to mutate several sites on 4E-BP1. Serine 83, serine 112, and threonine 70, and observe the effect of these mutations. In inducing these mutations, we want to decrease mTORC's ability to inhibit 4E-BP1 via phosphorylation, and then observe the effects on 4E-BP1's binding affinity for EIF4E. This project is a continuation of Andrew Verdesca's from last year, but with some key differences. Last year, Andrew wanted to mutate only one site on the protein. We want to look at 3. This is for a couple of reasons. One, it's been suggested by our mentor, Philip Geter, that mutating multiple sites on the protein will yield better results. And these are also sites that haven't been looked into by scientists before. This is legitimately an experiment that has not been done. The other key difference between our and Andrew's project is that we've put a little more thought into how we're going to test relative binding affinity. We've developed a concrete plan with Philip, and we'll continue to do so. Let's get into how we're going to run this project. So, I've separated the project into 3 main phases. Isolate 4E-BP1 and EIF4E, create the 4E-BP1 mutants, and then test the relative binding affinity. So part 1 is where we are now, in isolating EIF4E and 4E-BP1. We have to PCR 4E-BP1 and EIF4E, we have to purify, we have to perform restriction digests, we have to ligate, we have to transform the plasmid into cells, we have to inoculate, we have to mini-prep, and then eventually we're going to get to the point where we can send those genes off to sequencing. And we're going to use that sequence of the genes to determine our site mutation.
Where's 4E-BP1 and EIF4E now?
EIF4E now, I'll get to that when I'm in the data review. That's okay. So after we isolate the EIF4E and 4E-BP1, we want to create the 4E-BP1 mutants. And we're going to do this by G-site directed mutagenesis, which is basically running a PCR reaction with mutants in your primers. We're going to include the desired change in PCR primers, and we're going to test 3 different sites of mutations. As I mentioned before, the sites are threonine 70, serine 83, and serine 1112. We're going to change all of those sites into valines.
What's G-site-directed mutagenesis?
G-site mutagenesis is the process where you introduce a mutant in a PCR reaction.
What's the G? Does any?
Just a second.
Oh,
It's the same thing.
okay.
There's no G.
Sorry,
It's okay.
that was in directions from Andrew last year.
I don't know where to move that.
It's fine. It's fine.
Okay, sorry. And we're going to, ideally we can test these mutations 1 at a time, 2 at a time, and hopefully 3 at a time, but that's going to depend where the mutations are located on the genetic sequence. Keep in mind, these are sites on the protein, they're not the actual codons, so that's why we need to obtain the genes first so that we can match the codons to the amino acids. And then we'll use SOC and DH5 alpha cells to express the mutants. So this is the part where Dr. DeCilio, I, and Sophia have put in the most work as we've been meeting with Philip and figuring out ways that we're going to eventually test relative binding affinity. He suggested 2 methods that we can see whether our mutants worked. The first is an IPE assay on 4E and BP1. This might be a little bit confusing, but we're going to tag 4E with an HA tag and overexpress BP1. We want to see if when we pull 4E down, BP1 comes with it. If the 2 come down together, that would mean that they're bound. And if 4E and BP1 are both bound, that would mean that the protein's functioning as it normally should. In other words, it hasn't been phosphorylated, it hasn't had a conformational change. If our mutants if phosphorylation is occurring though, there's going to be less binding of 4E and BP1. That's purely concerned with the protein. One day we'd like to test in vivo in CHO cells, but we're going to need a lot more supervision and guidance with Western blots with that step. And I'll get to, we're at a very preliminary phase of this project, so I will talk about where we are now, and then we can plan more for the CHO cells later on. But here's where we are now. So since the start of the project, which was last year, we've been able to obtain the gene coding for 4BP1, and we've managed to reconstitute the plasmid containing it. But the vector containing 4BP1 isn't the one that we need, so we need to, using PCR, copy 4BP1 out of the plasmid and into a new vector. We've had mixed results with this, and we haven't been able to proceed past that PCR. This is a gel of a successful PCR reaction, but the band is really rather faint, and it's one of the only ones that have succeeded, but we do think that we can get this PCR to work, and we've been talking with Dr. DeCilio, and we were starting to get better results towards the very end of last year.
2 minutes.
This is on the 4BP1 side. On the EIF4E side of things, Bridget last year was able to successfully PCR and PCR purify EIF4E as well as mini-prep the expression vector PTXB1. Our next step would be to digest and ligate EIF4E into the expression vector PTXB1. Now, logistics-wise of continuing this project, we have mini-prep stocks, we have plasmids, we have primers from last year for the wild-type 4BP1s and EIF4E. We're going to need to order new primers for the mutations as our mutation sites have changed, and then if we one day want to test in CHO cells, one cell line from Sigma Aldrich runs $400. I calculated this number using about like a $30 per primer, which is pretty generous. So I'd say overall for the next 1 to 2 years, estimated cost of $600 for this project. A lot of the procedures we do are things such as ligation, PCR, where we have the kits in the lab. So our mentor, as I mentioned, is Phil Peter. He came here. He's an NYU doctoral candidate, and this project was taken from research that he's been doing. Our supervisor is Dr. DeCilio, for anyone who's interested, and so far it's just me and Isabella working on this project. Isabella is an AP Bio student, and she is interested in what we're doing, and she's going to help me run lab procedures as part of her AP Bio project. In terms of taking on new people, I'd love it if you guys had taken Research 1 or plan to take it, but that's really an encouragement, not a requirement. And as Isabella and I both have after-school commitments, you should know that our work hours are normally after those. So for instance, after winter track practice, like 4:45 to 5:30, 2 to 3 times a week. And we do a lot during flexes and on iRT Thursdays, at least we plan to. And we meet during Monday CP. So please let me know if you're interested, or let Mr. D know. Hopefully this gets approved, and thank you for listening.
exactly 10 minutes.
How's everybody looking?
Because do you want us to go?
I think your costs are off. So I just, it doesn't have much to do with approval of the project, but if you're gonna grow CHOs here, that's gonna be expensive.
Media costs.
The media is gonna be quite a lot of money.
Okay, to grow them here.
You
Yeah.
also might be able to, you might not have to buy the cells too, there's a possibility of getting them
Okay.
from somewhere else.
Okay.
But the maintenance of them.
But
Yeah.
That's
you're gonna, I mean, like, I imagine you're gonna contaminate them the first few times. It's gonna be,
We're a bit away from that.
That's why the Trosel, I mean, again, we're on phase 1 of the project. If we go back to these 3 main goals and relative binding affinity, the CHO cells is kind of like a last step that would be so cool if we got there. So we're very far away from that. I want to like have high hopes, but I'm not sure if we're going to get there this year.
okay, so EIF4E, so you have EIF4E basically ready to express, right?
Yes,
Um,
basically.
What's the problem with 4EBP1?
We weren't getting PCR reactions to work, but we found that we were using, Andrew and I were both using slightly different protocols, and then once we started using the same one, it started to work better. So another thing, that 4EBP1 is very small, and it might not, like, you have to remember, it's a like small gene, so the band size isn't huge, and you're not going to get a ton, and you're not going to get a ton nanodrop concentration-wise, so we might have been over-pessimistic in interpreting our results, but regardless, we don't think it's like, we think that we're going to get it pretty much.
If you spent a year trying to get 4EBP1 out of one plasmid, might it make sense to get it somewhere else?
We could try that, but as I said, towards the end of the year, we were starting to see better results, but this was past the time of data review and presentation and more towards wrap-up. Additionally, Andrew and I didn't really start doing lab work on this project till like December maybe of last year, well past November.
Right.
So, and before that, there were mini preps to do, so I recognize that it seems like a year, not a lot that happened, and that's kind of true, but.
No, I'm not suggesting that you guys didn't work. I know you guys worked. What I'm suggesting is that
No, it wasn't that, but just that like a lot of effort on one hiccup. For a while we thought that we got it to work, and then we tried to replicate the reaction, and then that didn't. So
where you guys are now is that you have it PCR'd, but not
some of
sequenced.
it PCR. We want more PCR.
But you haven't sequenced it.
Correct. Correct.
And what do you suspect the three mutations are going to do to binding?
So we're changing the binding sites into valings, which are sites that can't be phosphorylated due to their structure. So we're hoping that if 4EBP1 can't be phosphorylated to mTOR, it's not going to have that conformational change that makes it unable to bind to EIF4E. We're hoping that through ablating phosphorylation, we get better binding and better sequestering of EIF4E.
Cool.
I don't know.
All right, next one.
Thanks for the love and support.